We just continue our discussion of muscle physiology with cross bridging. It's the third event in contraction after the excitation and the action potentials and all that stuff. Once the calcium is released from the sarcoplasmic reticulum, the calcium, which is now in the sarcoplasm, binds to troponin. And this is the trigger for what's called cross-bridge cycling. It's part of the sliding filament theory of contraction. So if you remember the last semester, we talked about configurations of proteins, that there were primary structures, the amino acids, secondary structures, the folding or helical nature. The tertiary structure is the folding and the shape of the protein. And if more than one chain of proteins is together, that's the quaternary structure. Well, troponin has a quaternary structure. It's three globs that are intertwined and interconnected, but each one of those three globs has a tertiary structure. And if we cast back and remember this, the tertiary structure depends on the temperature and the pH and the electrical nature, the polarities of the R groups in the amino acids, forming bonds with other R groups. Calcium is a very strong cation. It has 2+ charges. So the troponin tertiary configuration with calcium is a different electrical charge than without the calcium. So when calcium has bound, the troponin changes shape. And if you remember from the previous video, troponin is bound and it has a binding site with actin and with tropomyosin. Tropomyosin is the long rope-like structure. So when calcium binds to troponin, the troponin changes shape and it moves the tropomyosin in relation to the actin because it's bound to both. So one shape without calcium, a different shape with calcium. Now, again, we have to remind ourselves of a few things, but the myosin head has a binding site for actin, and the actin bead has a binding site for myosin. When these two things join, when a myosin head attaches to the exposed binding site on actin, we call this cross bridge. And this happens because normally at rest, the tropomyosin covers the binding site on actin. When troponin changes shape, it pulls the tropomyosin out of the way and exposes the binding site. And binding happens. Now, myosin, when it is in a high energy configuration, the head is extended. When the potential energy stored in the myosin head is turned into kinetic energy, and this happens when ADP and P are released. Then the myosin head moves into a flexed position. So ATP attaches to the myosin head, gives its energy to the myosin head, which puts it in an extended position. When the ATP and P leave, they disengage, the myosin head moves into a flex position with that energy that was transferred, turning into kinetic energy and moving the myosin head. When the myosin head moves into the flex position, it pulls the actin that it is attached to towards the midline. The myosin then stays attached in the flex position until another ATP binds to the myosin head, causing it to release from the actin and move back into its high energy configuration of extended. Okay. So the ATP splits into ADP and P and transfers that energy to extend the myosin head. So if we look a little bit closer, we're going to see that in a relaxed position, the thin filaments and the thick filaments overlap somewhat. The H zone is quite large. The I band is also quite large. There is no cross bridging happening between these things. This is what it looks like in this relaxed position. The head is extended here, which means it's high energy. It's bound to ADP and P. The ATP is given the energy and put it into this configuration. So this is what we have. The troponin here has bound to the calcium and pulled the tropomyosin out of the way, exposing the binding sites on the actin. The binding site here on the myosin head is lined up with the binding site there on the actin, and so this happens. It binds. The myosin head can't be bound to ADP and P and actin at the same time. So as soon as that bond is made, then the ADP and P leave. When the ADP and P leave, the head moves from its extended to a flex position, pulling the actin molecule along with it, pulling the thin filament towards the midline. And it will stay that way until an ATP attaches. Now, the myosin can't be attached to actin and ATP at the same time, so it disengages. When that happens, the ATP becomes ADP and P and gives its energy to the myosin head, which goes back into its high energy configuration. And this repeats as long as calcium is pulling the tropomyosin out of the way. And you will see that what ends up happening is all of these heads with the cross bridges have pulled the thin filaments towards the M line, towards the middle of the sarcomere. This is quite a dramatic one. The H zone is virtually gone, and the I band is virtually gone. And so what we end up doing is we've pulled this Z disc and this Z disc closer together because we've pulled on the thin filaments. The A band stays the exact same length, because it's the thick filaments. So this repeats as long as the calcium is there and as long as there's ATP. If there's no ATP, then the myosin head can't disengage at this step over here. This is what happens when you're dead, because dead is the only time we have no ATP. This is rigor mortis right there. It doesn't disconnect. Take the calcium away by pumping it back into the sarcoplasmic reticulum. Then what happens is the troponin, or the troponin, if you would, changes shape again, and the tropomyosin reblocks the binding site. And when the binding site is reblocked, then the cycle is broken. No more cross bridges, and the sarcomere relaxes and lengthens again. You'll notice that none of the filaments change length. They just overlap more when there's a contraction. They overlap less when there is relaxation. The filaments slide past one another, hence the sliding filament theory. So as long as there's calcium and ATP, the Z discs move closer together. The H zone and I band narrow and they can almost completely disappear. So if we look at photographs of this, we can see that in a relaxed state, the A band is from here to here, big H zone, the M lines here, the Z discs are this far apart. The same sarcomere contracted, the H zone and the I bands have completely disappeared here, and the thin filaments are meeting at the M line. So this is fully contracted, that's fully relaxed. We won't get into neurotoxins and things like that, right? But this is what happens. Okay, so for muscle relaxation, the nerve signal at the motor neuron stops. So there's no more release of acetylcholine. I told you about acetylcholine esterase being present in the synaptic cleft. It hydrolyzes the acetylcholine, breaks it down, makes it no longer functional. So then the ACH receptors, the acetylcholine receptors, close and sodium stops coming in. So there's no further action potential. No action potential. The calcium channels in the sarcoplasmic reticulum close, and the calcium is pumped using ATP back into the sarcoplasmic reticulum, which removes the calcium from the troponin. The troponin returns to its original shape. The tropomyosin then blocks the binding site, and the muscle, no more cross bridges, the muscle elastically returns to its original length, its resting length. Okay, so we've been talking about ATP. ATP having a number of jobs to do. One of the jobs it has to do is run the sodium-potassium pump to reset the resting membrane potential at the sarcolemma, to put sodium back on the outside and reset the trap, reset the trigger. Another thing ATP has to do is the calcium pump, to pump the calcium back into the sarcoplasmic reticulum and take it away from interacting with the troponin. It also is used to give energy to the myosin head. And as long as the myosin head is cycling, it's using a lot of ATP. So there's a whole bunch of ATP necessary for a muscle contraction. Now, there is some ATP stored in the muscle itself, in the cell itself. It's made at times of rest and is stored up. The problem is that there's only about 5 seconds worth of ATP stored. Hardly any amount to keep that contraction going. 5 seconds, not very long. But there's 3 ways that we generate more ATP. The first way is creatine phosphate. The second way is glycolysis, which you remember only gives us 2 ATPs, and lactic acid. And the third way is aerobic respiration in mitochondria, where we get 36 ATPs and carbon dioxide and water as the byproducts from one cellulose, or one glucose, rather. So the first thing we'll look at is creatine phosphate. So creatine phosphate, literally creatine, when there's all the ATP is made, when all the ADP is turned into ATP, then any extra energy being formed, being converted, gets stored as creatine phosphate. The phosphate, instead of adding to an ADP, adds to the creatine. It's a high energy bond. As soon as there's ADP available, the P will leave the CP and turn the ADP into ATP. The phosphate would rather be in ATP than in CP, in creatine phosphate, right? If you're loaded up with creatine, you have enough phosphates bound here to instantly create ATP for about another 10 seconds of additional energy. But 15 seconds total is kind of the going thought. So in a sport like the 100-meter running, the person can do the 100 meters on their stored ATP and their stored creatine phosphate. But that's about as far as you can get. In the 200 meter, so the 100 meter record is 9 point something seconds. The 200 meter isn't 19 seconds, it's more like 21 or 22 seconds, because there's not enough ATP and CP to run the 200 with. We have to regenerate ATP in a different way. The fastest way is glycolysis. It's when we get the glucose breaks from a 6-carbon chain to 2 3-carbon pyruvates. We get 2 ATPs out of it. It happens in the cytosol, and there's no oxygen involved. It's just a couple of steps, and you end up with 2 ATPs. The pyruvates, if there's no oxygen available, become lactic acid. They become lactates instead of pyruvates. And we get a couple of ATPs very quickly. So if we have a lot of glucose available and no oxygen, this is the way that we get ATP. Gets us that last 100 meters in a 200-meter dash. This gets us, so you can get to first base on stored ATP and CP, maybe to second. But to get to third base, you're going to need glycolysis. Home run, you can slow down, so you have more time. But a triple requires this. Okay, if there's oxygen, then the pyruvate ends up in the mitochondria, and we oxidize it, we mix it with oxygen, we get carbon dioxide, we get high energy molecules like NADH and FADH2s, which we've talked a little bit about, but we generate some more ATP. Right? We can run fats into aerobic respiration, and we have a lot of ATP because the fatty acid chains have way more than 6 carbons in them. And it's the carbon-carbon bonds that provide the energy. Okay, so within the muscle, the first 5 seconds stored ATP. The next 10 seconds or so stored CP. The next place we get it is anaerobic. We get a couple of ATPs. And if we can get enough oxygen delivered, we can get 36 ATPs out of, we're going to get over 30. And it varies depending on pH and a bunch of things. And that's plenty of ATP to keep the muscle contraction going. Okay. If there's no oxygen or low amounts of oxygen available, the pyruvate becomes lactate, lactic acid, and the lactic acid is what changes the pH, causes some pain, but it can be used as fuel and brought back into brought back in and make the ATP 5 minutes after you finish the race. Some of it is going to into the blood and go to the liver, where it gets turned back into glucose. And then we have to transport the glucose back to the muscle. This takes a lot of time. So if we're sprinting, the ATP, right, you can get the energy that you need from phosphate transfer systems, from the stored ATP and the creatine phosphate. If it's just a minute or less, we're primarily going to get it by glycolysis. And the reason that that's going to happen is because the whole aerobic cycle takes time, and the race is already over before you can supply that ATP. So we use this aerobic ATP for more endurance, like long swims, long runs, the bicycle racing, that kind of thing. So the Tour de France guys are all aerobic. Baseball players tend to be primarily anaerobic, right? Weightlifters or 100-meter guys tend to be all CP and ATP storage. Like Olympic powerlifting, huge amounts of ATP required for a couple of seconds. That's why creatine supplements are popular amongst weightlifters, because it will extend that period of phosphate transfer. Now, these are not hard and fast rules. We're going to be doing some phosphate transfers. We're going to be doing, it depends. So if you're running a marathon, you might be making ATP slightly faster than you're using it, in which case you start storing it again and storing it as CP, and then you'll be taking that out. And like, so these things all end up working together. Now, this brings us to a concept of oxygen debt. If the full aerobic respiration of to make the AP that you required for the event that you've just done. Say it was 10 liters of oxygen required. And you only had time to deliver 4 liters of oxygen during the event. That means that the other 6 liters of oxygen that would have been necessary if all of the glucose used was completely burned aerobically, oxidized aerobically, you actually owe it to your body. And we call this oxygen debt. This is why when somebody runs to third base for a stand-up triple, when they get there, they go, they're panting. And the reason is because they're trying to bring in that extra oxygen. They're replacing the oxygen that was used off the hemoglobin and the stored oxygen in the muscles, the myoglobin. We use it to replenish the glycogen, to replenish the ATP and the creatine phosphate, and to convert lactic acid back to glucose so that we can reuse it later. Oxygen is required for these things. And so basically think about the ways that we get oxygen or we get ATP without using oxygen and convert that to that much glucose being completely oxidized aerobically. And the difference is the oxygen debt. So I like to think of the oxygen debt as the oxygen required after the event to restore everything to its pre-event levels. Obviously, the more you can deliver oxygen during the event, the less oxygen debt you incur. And the more endurance you have. This is why Lance Armstrong did blood doping. By having more hemoglobin, he could carry more oxygen and reduce the oxygen debt. Literally getting into shape. If I was to go to the gym right now and work out for an hour, I would be in oxygen debt. But if I did this every day, what would happen is I would grow more red blood cells to supply oxygen more efficiently. I would grow more capillaries to the tissues to deliver oxygen more efficiently to the muscles I'm using. I would also grow more myoglobin within the muscle cells to store more oxygen within those muscle cells. And therefore, the same amount of exercise, I would incur less oxygen debt the more I train. That's what getting in shape's about, is getting rid of this issue. Okay. Okay. So muscles are made up of different types of fibers, and we can classify them, the fibers, the different types, based on two things: the type of contraction generated, uh, and that is power versus speed. So it's really fast and slow contractions. Uh, the other way we classify them is by the way we get ATP. So the power is related to how many myofibrils are in the muscle fiber. So it's the diameter of the muscle fiber. More myofibrils, the stronger the contraction. But the speed depends on the type of enzyme that that breaks the ATP that's bound to the myosin head. So quick ATPases allow the myosin head to go back to its high energy configuration quicker, and the slow ATPases is slower. There's other things about how quickly we can release calcium, and that depends on the number of channels, but how fast it reuptakes into the sarcoplasmic reticulum. All of these things are controlled by enzymes. And basically, that means that the ones that can do it quickly, we call fast twitch fibers, and the ones that do it slowly are called slow twitch fibers. Fast twitch fibers contract very quickly and for a briefer amount of time. Slow twitch contracts slowly for a longer period of time. We can have oxidative fibers, which means that we get most of the ATP using aerobic respiration, that oxygen is involved. Or we can get glycolytic fibers where the ATP is supplied by glycolysis. Very quick things. Now, the oxidative fibers, in order for them to have lots of oxygen, the first thing they have is they store oxygen within the fiber itself. So there's a large amount of myoglobin, and myoglobin is red. So these fibers tend to be red. Because they get their ATP aerobically, there's got to be lots of mitochondria. And there's extensive capillaries. There's a real good blood delivery network that's going to bring glucose and oxygen to the muscle very effectively. These fibers are really fatigue resistant. They're red in color. They produce a lot of ATP per glucose, and they can contract for a long time without running out of ATP. Glycolytic fibers use anaerobic cellular respiration. So they don't need mitochondria. They don't use oxygen, so they don't have very much myoglobin. Right? They don't have a lot of capillaries, but they have to have a lot of glucose stored in them, a lot of glycogen, glucose in the form of glycogen, because they're only getting 2 ATPs out of a glucose. So they need lots of glucose. And you can't deliver the glucose very quickly, so you have to store it in the fibers. These are called white fibers. Okay? They react with a lot of power very quickly, but they're quite fatigable. So colloquially, we call them red fibers and white fibers. Okay, I want to just kind of demonstrate this and have you think about it. Turkeys are big, heavy birds, and they walk around all day long. They're on their feet all the time. So their legs have to have these fatigue-resistant oxidative fibers as the predominant fiber in each muscle. Lots of capillaries, lots of mitochondria, lots of myoglobin, so the meat is dark, fatigue resistant. Turkeys don't fly worth a damn, and when they do, they don't go very far. So their breast meat has to have a lot of glycolytic fibers because it's going to create a lot of power in a short burst of energy. And turkey breast meat is white. It doesn't have very much myoglobin in it, and it's starchy. It's got lots of glycogen reserves in it, and there's very few blood vessels in it. This is why turkey breast is white and has that starchy taste and not very many blood vessels. Isn't very moist. The leg meat, the dark meat, is predominantly red fibers because of the endurance that the bird needs during its life. If we contrast that with duck meat, another fowl, ducks fly long, long distances when they migrate. They need a lot of fatigue resistant fibers in their breasts. Duck breast is a red meat. There's and they don't walk very much. They float. So their legs don't have the same amount of oxidative fibers. That's just a very easy way to think about this. Okay, so if we combine these two things, the fast and slow and the oxidative and glycolytic, we could end up with 4 types. However, slow glycolytic makes no sense at all because glycolysis, the advantage to glycolysis is that it's fast, and that would be offset by the slow ATPases and the slow contractions. So there is no such thing as a slow glycolytic. But slow oxidative fibers make a lot of sense. These are high endurance, aerobically charged, red, myoglobin full, mitochondria full fibers. These would be your postural muscles. These would be your gluteus and muscles like that. They're the type 1 fibers, slow oxidative. Fast oxidative fibers are intermediate. They're fast fibers, but they still have a lot of mitochondria, and they primarily get their ATP from aerobic means. But these are intermediate in terms of power. These so these are intermediate in terms of endurance. Right? So they tend to be light red in color. They have less myoglobin. The fast glycolytic fibers, you get fast and powerful contractions. The contractions are brief, they're large fibers because they're so powerful. You don't need to diffuse oxygen into them, so they can afford to be large, and they lack myoglobin, so they're white in color. So these are the fast glycolytic fibers. So turkey breast meat is fast glycolytic, and turkey leg meat is slow oxidative, predominantly. Now, that doesn't mean that a muscle can't be some mixture of these things. Like, so a muscle like your biceps will probably have some slow oxidative that you will use when you're carrying a suitcase. They will have some fast oxidative that you're going to use when you're just doing repetitive things and just using the muscle. They'll have some fast glycolytic fibers when you're swinging a baseball bat or a golf club or whatever, like you're doing something fast and powerful. Now, every person will have a mix of these three types of fibers within any given muscle. The relative frequency of them varies. So some people are better suited to fast, powerful things. Some people are more slow oxidative fibers. But I will submit to you that the sport that you choose, you will exercise and develop and get hypertrophy of the types of fibers that you're using the most. So if you decide to take up weightlifting, especially powerlifting, the fast glycolytic fibers are the ones you're going to be using the most, and so they're the ones that are going to hypertrophy. And you're going to bulk up, and you're going to have these things. If you take up marathon running, you're not going to bulk up. You're going to have the high endurance. You're going to be using and developing the slow oxidative fibers. So all of this stuff depends on what you're doing. Now, one of the things that I will point out is that you can't turn slow oxidative fibers into fast glycolytic fibers. It doesn't work that way. It's different enzymes, it's different things, but you can optimize them. So if you are optimizing your slow oxidative, you will grow more mitochondria. You will make more myoglobin, and you will make the slow oxidative fibers more efficient. If you are weightlifting, you're going to hypertrophy the fast glycolytic. Okay? So any muscle is a mixture of these types. But depending on what you're doing, different muscles have different mixtures. So muscles for your hands, lots of fast glycolytic because you want quickness of movement, playing piano and that kind of thing. Back muscles, slow oxidative to maintain postural support. People that are long distance runner have a higher proportion of slow oxidative fibers. Is that because they've trained those fibers, or is that because they are born with more of those fibers, which then makes them want to take up that sport? It's probably a little of both. Sprinters go with more fast glycolytic. So if you look at a particular muscle, even within a fascicle, you see that there are all three fiber types represented. Okay, so let's end this video here, and we will pick up at muscle tension in the next video.